Thank you. So I'm not going to talk about anything from the Ralph et al. Papers. I'm going to be talking about lots of other things, all of the new stuff that's exciting me. So today what I want to talk about is the interactome. Now, the interactome is an opportunity for us to get new information and new insights. So the description of the interactome is a whole set of molecular interactions within a cell. Now, I'm going to broaden that out to tissues, whole plants, and I suppose even for the audience, because there's going to be a lot of ecologists here, even to communities, but I think the stretch might be a little bit far. Now, for those that know me, I'm a reductionist. I'm very proud of being a reductionist. I enjoy understanding process and what makes the plants tick. I suppose Ralph et al. And all of those earlier papers was all about where I was fascinated by understanding how the plant worked, and I used a particular technique, chlorophyll fluorometry, as the tool. What I'm going to talk about today is how I use and anchor a lot of new molecular tools to fluorometry, because fluorometry is a bunch of smoke and mirrors, and Sven and I have had mic up higher. That better? Not much? Can I turn it up or yell more? Yell more. Okay, yell more. Oh, Jesus. Smoke and mirrors, Sven and I. Fluorometry is an opportunity where we can probe what happens inside a cell, and we think we know what's happening. I've been fascinated since the two ISBWs, the one in China, where I'm using new techniques, or the group's using new techniques to inform what we think we're seeing in fluorometry. And we've got another set of smoke and mirrors. We've got this molecular tools. So I'm bringing together tools. I want to anchor one set of data and measure and understand processes in the other. So that's where I come through with my mechanistic understandings. So what I want to talk about today is we're going to start out talking about genomics, databases, and PCR. We'll move into transcripts, and I'll talk about 3 light experiments, and that's a little bit of an evolution. So it's what we started out doing back in 2014, what we did in 2016, and what we're doing right now. And each of them, I'll show you where the problems were, how we got better and better over time. We'll also talk about an ammonium data set. I've only got 2 of those data sets. I'll quickly talk about secretomes, plant microbial interactions, because I think this is an undersung part of the process, and for those interested in blue carbon, this is critical. The biogeochemical processes and understanding how the plant, the bacteria, and the chemistry interact is very, very poorly understood. And I've got some early data or some suggestions. Then I want to move into the proteome. And so the proteome, the metabolome, and then on to systems biology. And then finish up with the idea of toolkits and data visualization, how we actually use all of this new information. So it's all about how the omics can be brought to bear on seagrasses. And I am absolutely not a molecular biologist. 5 years ago, I went to my university and sat in first year molecular biology 1 with all the undergraduates. Didn't do the pracs, but I listened to all of the lectures. And so I've kind of taught myself, and so what I'm giving you today is not a specialist molecular biologist. And for those in the audience that are hardcore omics experts, I do apologise. But for the broader group, what I want to do is I want to encourage you to start thinking about how you can use these technologies to inform your biology and take you to new steps. I'm really excited by where we've been able to go with this. So, miraculously enough, 4 years ago when I was over in China, I used the same backdrop, and my topic was Systems Biology Approach to Understanding Seagrass Decline. That was 4 years ago, and we were just on the edge of having the first transcripts, the first genomes, the first de novo assemblies. So we just started to have the information, and I was kind of excited, and I thought I knew where we might be able to go. This figure here is quite dated now, but I think this got us starting. This is understanding stress and mortality, and it's specifically about light stress. And you see how the fundamental change in the morphology changes over time, but the thing is, things like panfluorometry does not change quickly enough. And this is why, for dredging problems and a lot of these issues, fluorometry wasn't good enough. We had molecular responses back here, where we had RNA responses being very quick. I think we need to update this, and we now need to have a proteome response here and a metabolome response there. So these are intermediate between these. But back then, we didn't have the technology. And I didn't go to Wales, so I didn't get a chance to tell anyone where we're up to. Now, this is not new science. People in the audience here, Rue, Mats, Sven, Gabriel Poccaccini, have been telling us all of this since 2012, 2013. This is the roadmap that my labs used. So the seagrass community knew what to do. I think the problem was the expense and the technology wasn't available. I've been fortunate back at UTS to actually bring together a lot of those tools and experts that we've been able to practice and put into place what these leaders actually suggested we should do 5 years ago. So what I want to talk about is genomics transcriptomics, the challenges with bioinformatics, the secretome, signaling hormones, proteomics, metabolomics flux, chemometrics, and a little bit on the microbial interaction, all in 40 minutes or something. And Sudhi, if you can tell me about 20 minutes in or 30 minutes in so that I make sure I finish on time. I rehearsed this, I've been putting it together for quite some time. I rehearsed it this morning and it was about 1 hour and 10. So I chopped out a whole bunch of stuff and now I hope I don't finish on 30 minutes. So we'll see how it goes. So, how all this stuff fits together is really important. So, starting out across the bottom here. So the genomics, the transcripts, the proteomics, the metabolomics. We start out with DNA sequencing and we understand what structures the actual seagrass. Then we go along to RNA sequencing and we generate the transcripts. The transcripts tell us what's actually changing at the gene regulation level. Then we go up another step and we move on to the proteomics where we bust apart all of the peptides, reassemble them and work out which proteins have been changed and regulated. Doesn't quite tell us everything. It's another stamp collection. This is a stamp collection of genes, of regulated genes. This is a stamp collection of busted apart proteins. This is where we start to get into some things that we can get our teeth into, the metabolomes. When it's grouped together and it's got a pathway and we can actually understand why these compounds have changed. Now, it's not all about just collecting cool data with omics on the end of it and coming down to this trendy thing called system biology, which is actually quite a pain and quite difficult. What I'm interested in is these two things, the phenome of the plant, the phenotype, and also markers. How can I use these technologies to tell me about how the plant is functioning at its phenotypic level, and how can I use these new insights to guide my selection of biomarkers as a tool for management? So there's, in the back of my mind, there is a lot of reason why we're selecting these tools. We're not just doing it because they're trendy and fun. And they're very, well, they were expensive, they're getting less expensive these days. Okay, so to start the story, 2016, Gene Olson's release of the sequenced Zoster marina genome made the front cover of Nature. This allowed and pushed things forward very, very quickly for us. We were working, doing real-time PCR. We had the early data coming out of this, but understanding how our Zoster, which isn't Zoster marina, and Zoster marina function, there's still differences. So this was an important step that allowed our transcripts to be anchored and for us to actually understand how a properly assembled genome worked. We also had Positano oceanica. So this is Gabriel Poccaccini released this one, and this is another, so this is an EST-based de novo assembly, and what everyone does when you release a genome, you compare it to a whole bunch of lab rats. So you compare it to Arabidopsis, you compare it to Brassica species. It doesn't tell us a lot. Generally what we find with seagrasses is they're closest to vines. Why? That's not my area. But the thing is, having these de novo assemblies coming through has allowed us to move forward in the use of the genome data. Back at UTS, we had been chipping away at Zoster muelleri. We were working originally with Dave Edwards when he was up at UQ, then he moved across to the University of West Australia. Gary joined us on this, and this was the first Southern Hemisphere genome that was released, and so this was Zoster muelleri. This then allowed us to start seriously understanding where our real-time PCR, where our transcripts were actually telling us. And instead of us comparing to Zoster marina, we had our own genome. We did the usual thing. We worked out what it was different from Arabidopsis, Brachypodium. There's 650 conserved gene ontology terms with all of these other model higher plants. But that's not the most interesting thing for seagrasses. Seagrasses, because of their unique habitat, it's more interesting what they don't have. And so with Zoster muelleri, we started down the path of understanding that we'd lost one of our hormones. We'd lost ethylene. Now, looking back through the marina genome, it also corresponded that they'd lost it. Floral organ development, H2S and metal sensing. Now, this is interesting. You know, both of these compounds, H2S and metals, are very, very important to the roots of seagrasses. So perhaps we've lost higher plant lab rat versions, but I'm sure in our transcripts and in our assembled genomes we're going to find some very, very interesting data in that space. Loss of stomata, obvious. There's no need for the stomata. So we've now got 4 genomes: Zoster knoltii, marina, muelleri, and Positano oceanica. So we've got 4 genomes. A lot of data. How do we make that available for people to use? So there's a couple of databases that were put together. First of all, Dr. Zompo was put together. That only had Zoster marina and Positano, and that was an EST library. So you could go into it, mine it, and find if you had gene sequences that you could use. We used this originally for a lot of our early work. There was also a second transcript-based database that was released just for Zoster marina. What we did is we put together a database called Seagrass DB. It was released last year, and that brings together the data, the publicly available data that's in Dr. Zompo, and brings together a bunch of our transcripts, and when we sequenced a number of other seagrasses. So what we did is this is where we put our data. So we've got 120,000 unigenes of Zoster muelleri in the database. This is the publicly available marina, Positano oceanica, and then we put 6 others that we ran transcripts of. Now, those transcripts they're not covering a particular stress condition. We didn't do anything to the plants. They're just general growth conditions. As people do experimental work with any of these other species, it'd be great if that data was uploaded. Then we could have a larger database where we'd have more access to information. At the moment, I'd say our group, we've got some work going with Positano australis, not oceanica, and we'll continue putting data into Zoster muelleri. But as the database grows, it's going to be more useful to people. This was some work between an honours student, Reagan Hayward, and Gorev Sablok. And so you needed a bioinformatician and a web designer to actually build this, to actually make it effective. So it was quite a bit of work for the 2 of them. Quick insight into what you can do. You can blast any of your sequences that you've got. You can retrieve primer sequences that you might want to actually generate for real-time PCR. An example of how large it is, there's a screenshot of about 50-odd unigenes from Zoster muelleri, and there's 3,027 other pages in that database of that information. So it's a very, very rich source of information, but you've got to know what you're doing and how you're getting the information out. So publicly available, there's its URL down the bottom. Please use it and provide new data to it. One of the simple things you can do with it is you can extract gene sequences for developing primers. So there's one of the unigenes. It happens to be a ribosomal protein. That's its sequence. You can then generate primers, and you can put that data back into KEGG maps to understand which of the genes are regulated in the process that you might be looking at. We'll come back to KEGG maps towards the end. So we've now got a bunch of genomes available, and the standard tools that we were working with back in 2014, 2015 was this real-time PCR. So we'd try and identify a gene that we thought would be regulated for a process that we're interested in, and then look for how that could be used to inform our experiments, whether or not we're using microsensors, panfluorometry, stable isotopes. That's what I said before. I wanted to ground the data in something that's more robust and inform it with the real-time PCR data. So this is Matt Pernice. He's been with us now about 5 years. He's got a strong background in stable isotopes, and he's a molecular biologist. Now, Glenn put this scheme up yesterday, and you know, for people working with nitrogen assimilation and ammonium, that's a standard process. So we've got glutamate synthetase, and we've got glutamine, oxidoglutamate aminotransferase. Two enzymes, one that brings in the ammonia, and the second one that sticks a carbon chain onto it to make a product. So if we understand the activity of these two enzymes at a gene regulation level, that can then inform us how the plant is actually taking up its ammonium. So this is the data. So we've got stable isotope 15 linked up with the ammonia. We've got leaves and roots down the bottom. And all I want to show you here is that we've got a much higher uptake rate for the 15N for the leaves, and it's about a quarter the rate that we've got for the roots. Now looking back at the other side there, we've got our two enzymes. So we've got GS and GOGAT. And in the leaves, the genes for those two enzymes are upregulated strongly very, very quickly for both of them. Whereas in the roots, there's even higher upregulation, but then it's downregulated after about 30 hours. So something's different between the roots and the leaves, and that's evident in their uptake rates. So now we're using the regulation of genes to give us some insight into the mechanistic pathways that we're measuring as a stable isotope uptake. What we're going to, and I'll show you later on, the story of the ammonium moves on, and we've got some metabolomics data that we've used to dig more deeply into those pathways and understand what's happening. We haven't done fluxomics yet, but we certainly will be, and that's where you put a stable isotope in and you measure the changes, the rate changes of your compounds that are taking up the labelled isotope. Quick plug for Michael Kim. Michael Kim is going to be presenting tomorrow in the afternoon session, and he's going to be showing oxygen data with real-time PCR and understanding how the seagrasses function under low oxygen conditions. So this is another one of these types of experiments where we've integrated the real-time with an absolute measurement. Okay, this is the first of our low light stories. Now, this started back with Gladstone Port Authority. Mike Rashid and the group up at JCU were part of this project. And what we were trying to do was to develop markers to understand when to control dredging. Now, this was started back in 2014, maybe even 2013. And we had no Muelleri genome to inform any of our selection of primers. And so it took us an inordinate amount of time to get 3 housekeeping genes and 12 regulated genes, because we kept going to the Zoster marina, taking a sequence from Zoster marina and finding that it wasn't quite the same as Muelleri. So as soon as, and this is part of the evolution. This is part of what we had to do before we could get the fully sequenced, the transcripts of Muelleri. So what we're doing here, this is what the JCU crowd did. They put out frames on the seagrass meadows, and we tracked the change in light. Now, I'm only presenting 2 of the gene sequences that we identified as being responsive to this trigger from going growth in normal light to low light and then back. So PSAE number 2 is a photosystem 1 enzyme, and you can see that as soon as you put it under low light, the gene regulation ramps up, and these are fold changes. So it's quite a significant fold change. As soon as you put it back into normal light, that gene regulates back down. So we've got a tipping point. We've got a marker gene that under certain light conditions, if we see that change, it tells us something. ELIP, early light induction protein, is another one that worked for this. So as soon as we put it under low light, it upregulated. It downregulated while we were still under 6 to 8 days under low light, but then when we returned it to high light, it didn't upregulate again. So these are 2 genes that allowed us to understand when a seagrass was suffering low light stress. That was back in 2014. Now, Matt's using a new instrument called a nanostring. Instead of us struggling, taking about a year and a half, 2 years to find 20 odd genes, he's not going to run 800 genes because we don't need to run 800 genes, but we've picked about 50 of the photosystem genes from Zoster Milleri. We've gone back to the transcripts, we've loaded it up into a chip now, so for one chip we can put it straight in, we put 48 samples, and in a day we've got the entire gene regulation of those genes that we put in. This is an extremely powerful new technology that you can work with. It's also an awful lot cheaper. So we're running about $500 a sample per lane for the transcripts, whereas we're down at about $140 a sample. And this is going to allow us to get infinitely more insight. Now that we know which genes are regulated, if we have a suite of 50 and we can run it in an hour, this will allow us to be very much more efficient. And this is what we're looking for, how we move towards toolkits and environmental assessments. Okay, so that's the end of real-time PCR. Now, 2015, we'd got to the point where we had the Milleri genome sorted out. This is where yesterday Naseem presented some of her transcript data. Now, I'm not going to steal her thunder. Now I can talk a little bit about what I think the opportunities with Naseem's work was. 20 years ago, when I started playing around with fluorometry, I used higher plant information to inform the discussion about metal contamination to seagrasses. And the accepted wisdom was that photosystem 2 was damaged. And I was using a fluorometer, and I found that quantum yield and non-photochemical quenching and a whole bunch of processes were damaged. So my inference was that it was photosystem 2. I love the fact that we've got a new tool. Fluorometry is smoke and mirrors. Absolutely. We've got 3 parameters coming out of a bit of light coming off a plant tissue. How can it be prescriptive to every last process? Now what Naseem's got is she's got controversial evidence. She's got no impact, no gene regulation on photosystem 2. Doesn't mean there's no damage, there's no impact, but she's got a lot more of a smoking gun on photosystem 1. Our lab's got a lot of capacity for doing spectroscopy and optical processes, and we'll do a lot more work on what she's identified. But this is the power of bringing together these 2 types of data. So, Naseem, you're on a winner, and I look forward to when she actually gets the metabolomes out and we can actually understand where our photosystems are. Okay, the second light experiment. First one, we did the best we could. Second one, Peter Davies showed up just at the right time when we had our first transcripts. So he became the owner of the transcripts. So the experiment that he ran, he put, you see that's pretty common, we don't change too much. Control light's 200 microeinsteins, low light's 20 microeinsteins. 6 days under these conditions, we measured the full suite of pigments. We measured a whole bunch of fluorometry. We understand how the biochemistry of the plant's going. Then Peter took transcripts of only the leaves, broke it into 2 groups, ran the bioinformatics, used Seagrass DB to inform his selection of where the transcripts went. Just quickly, the fluorometry data. Before treatment, none of the groups of shaded and control are different. So this is maximum electron transport rate, minimum saturating irradiance, and initial quantum yield. So all identical before treatment. As soon as you put them under you drop the light, your electron transport drops, your minimum saturating irradiance drops, and there's no change in the initial quantum yield. Surprise, surprise. Quantum yield doesn't tell you a lot. We've got photobiology now. We've got the pigments. So we've got a whole range of xanthophyll pigments. We've got chlorophyll A, B, we've got the total chlorophyll. So the control and the low light on 2 days versus 6 days, you can see that neoxanthin, there's a significant increase under low light conditions. Similarly with the beta-carotene, similarly with the lutein, chlorophylls as well. So lots of fundamental data to help us understand what happens with our gene regulation. We got back over 20,000 unigenes that have been regulated, and it was now a problem of trying to make a story out of that. We found that chlorophyll binding was important. We also found that red-blue wavelength shifting is important. Now this you've got to go back to wheat and rice. So the sensing of a plant, how close it is, it measures the relative red to blue wavelengths. When plants are planted a long way away, they don't have the reflected signature from each other. When they're closely packed, they're like this. So this is really interesting for seagrass meadows. So as a seagrass meadow dies, you can sense the plants, there's nobody beside me anymore, so the red to blue shifts are important. And so this is what we found when the plants were light limited. Chlorophyll AB binding is putting more antennas on top of the chlorophyll so that it captures more light. So heat maps. So day 2, day 6, this is comparing the control with the shaded on day 2, and then similarly with day 6. We had 500 genes were upregulated, 1,000 were downregulated. But the important thing is which ones they were. So we had photosynthetic genes, pigment composition, surprise, surprise, light reception and signaling, but then we had ABA, so we had another hormone pop up. So why was a hormone responding to shading? That became a smoking gun that we were interested in. But I think the nice thing is all of this gene regulation, we've got plenty of evidence that boring pigments, fluorometry supports it. Okay, so this is the next challenge, is how you take this gene regulation and make a conceptual story. You have to make cartoons. Now, most of you in the audience aren't excited by the Z scheme, but just quickly, this is the Z scheme. So photosystem 2, photosystem 1, electron transport through the thylakoid, cytochrome B6F. Now, what you have to look for is these little boxes. So a green box means it's upregulated, a red box means it's downregulated. Everything with photosystem 2 is downregulated in low light. Chlorophyll AB binding, some genes are upregulated, some are down. Interesting over here, the Z scheme. So NDHM is upregulated. Why might that be? Because we've got more cyclic electron transport. We're actually moving electrons. Instead of it going in a linear way, we're going in a cyclic way. Down here, photosystem 2. FTSH1 is a D1 protein breakdown gene. So this is where we're busting apart the photosystem 2s. All of the rubisco is downregulated. The inorganic carbon is downregulated. Photosystem 1 is downregulated. So this is how we've got to kind of draw our stories about what's happening. I'll talk about ELIPs again later. So that's also downregulated. Then into the photosynthetic pigment pathways. So we've got the pathways for making the carotenoids and all of the xanthophyll pigments. You can see they're all downregulated. The xanthophyll cycle is downregulated. The pigment systems, the chlorophyll metabolism is all mostly downregulated. But then down the bottom here, we picked up ABA again. So ABA was impacted by low light. There's a blow up of the system. So we've now got the gene that forms ABA is downregulated. The gene that's linked with the breakdown of ABA is upregulated, and the receptor of ABA is downregulated. So now we've got a hormone that breaks down sugars. And so what we're thinking is happening here is as you reduce the light in the plant, it actually goes, I'm running out of carbohydrates. I've got to go into my sugar stores and catabolize them. So it makes sense, but how would we have seen that with fluorometry and the standard optics that we had? So this is giving us new insights, and this is allowing us to go back into the system to understand more about hormones. But we've still got cartoons. We don't have an analytical way of bringing our data together. This is where I have my two hats on. So I run a program in microalgal biotechnology, and I also run the Seagrass group. What we're doing with microalgal biotechnology is we're doing the same things, but we're using some very cool new tools for data analysis, where we've got a metabolome data, we've got a physical data set, and we're doing pattern recognition. Now this is with a guy called Dave Kramer, Michigan State University. And this is one of the more powerful deep learning algorithms that we can bring together disparate data sets and find where the stories are. Instead of making cartoons, we're going to actually have relationships. And I think this is the next step for us to be informing how we use these omics data. Okay. I'm doing okay. I better speed up. Okay, the secretome. This is the part that I was going to drop out, but briefly, I think we're not considering how the plant's interacting with the bacteria. The rhizosphere, where all of the communication happens. There's techniques that we've been working on at being able to isolate the compounds that the roots actually exude. So this is the root exudate on an NMR, telling us which of the sugars are being exuded out of the roots under high light and low light. You can see there's different peaks and there's changes in the peaks. We put that into the mass spec and we get the same thing. This is under high light, this is under low light. You can see we've got changes in what the plant is exuding. Now we need to stick the bacteria into the system. This is some work by Vincent Schultz. We had these tubes where we stuck the roots, we collected the exudate we manipulated the conditions of the plant, and then we measured what happened here. And we got infinitely more bacteria in the systems that had a lot more exudates. Valentina's been doing a whole bunch of metatranscriptomics, and this morning when I put her data in, it crashed my entire presentation. So I apologise to Valentina, and I'll just briefly explain what I think we're going to be doing with her data. We're going to be bringing metagenomes, so we're going to actually understand who in the bacterial consortia fits in here, and then we're going to do metatranscriptomes to work out why they're doing it and what they're doing. So we're going to get function as well as peoples. What's that time? 20 minutes, okay. So this is going to be an important part of understanding the bacterial interaction, the interactome. Now, well, if I've got 20 minutes, I'm going to do okay. So, the proteome. The proteome is now what is the plant doing as far as protein regulation. We need the methods of doing proteome analysis is not optimized for seagrasses. So unfortunately, we've had to do a whole bunch of optimization for extraction, separation, and detection. There's now a couple of papers out, so it's much easier to do that. We've probably spent the better part of a year and a half getting that worked out. I'm going to show you two data sets, application to light, so this will be our third low light experiment, and I'll show you another one with ammonia. So, for those that aren't aware of proteomics, in a nutshell and really quickly, you run a 2D gel, you cut your spots out, you digest them, you bust them into peptides, you run them through a mass spec. So this is a liquid chromatography mass spec. You get fragments of your proteins, you get mass to charge versus intensity ratios. You look for these patterns in a database. The bigger the database, the better you actually come up with your peptide sequences. Then you use chemometrics, which is another version of bioinformatics, to identify your protein. So this isn't cheap, but I think the prices are coming down for that. And absolutely, you don't have to have an LC-MS on site to do this work. You outsource it all the time. We just have the luxury. I suppose for method development, it's good to have it on site. Once it's a routine tool, it's cheaper to get it done outside. So this is what Zhang did with Manoj, and I apologize that I don't have his photo up the top here. But he was working with Manoj for the better part of 6 months, and they did the method development. So this is that same 2D gel. So you separate it on size and on charge, and you've got hundreds and hundreds of spots there, and you cut out an individual spot, so that's spot 6. You run that back through the mass spec, you get your peaks, as I mentioned before, then you put it through the database to do your ion matching, and then your peptide matching. So there's a bunch of bioinformatics, but it's getting automated, and there's pipelines that we're certainly developing to make it easier. We've also done it for Positano australis as well. So we've got 2 extraction methods, and we've got proteome analysis now working. Problem with Positano australis is it's difficult to get permits to collect it because it's so rare in the eastern coast. So this is Manoj. Manoj has been working with us now for about 4 years, and he's leading the proteomics and metabolomics work that we're doing. So what he does, he runs a 2D gel to separate the proteins. We're now moving on a gel-free technique, and that's what Nassim's going to be using. So it's much quicker and easier. You don't have to cut your gels apart. You run your samples on the LCMS. You come up with an accession number for your protein using the NCBI database, and then you work out who's been upregulated and who's been downregulated. Same again, we've got our anchoring data, so boring fluorometry. We've got same light intensities, 20 microeinsteins is low light, 200 microeinsteins is control, and in this experiment we've done supersaturating light at 600 microeinsteins. So we've got both sides this time instead of just low light. There is the control. There's no difference between T0 and T10 between the electron transport rate and the minimum saturating radiance. Surprise, surprise, when you reduce the light, you reduce your ETR max and you reduce your minimum saturating radiance. And when you do the converse, you get the opposite. So the supporting data is there. I apologize for this slide. It'll get cleaner as I press the button. First of all, there's your differential 2D gels. So there's supersaturated light and control light. Now what we have to do is we have to work out where a spot has appeared that wasn't there before, where a spot has got bigger, or where a spot's got smaller. So that's for the bioinformatics work. What we did find was that 93 of the proteins were differentially expressed. So what we have, and I'll use this a couple of times, upregulated is this brown colour, downregulated is red, and new proteins are green. So looking around this chart, looks like you've got a mix of upregulated, new, and downregulated, because this is the highlight response. So we break them into these groups now. So this group here is energy, carbohydrates, and metabolism, basically. This one is antioxidants. This one is photosynthesis. This is gene information. So when you expose the plant to high light, most of the protein regulation is happening with energy and metabolism. Interestingly enough, a lot of antioxidant activity is happening. Then there's a bit of photosynthetic work, and then it gets smaller as it goes on. So of the 93 genes, 47% were upregulated. So we've got pyruvate reductase has been upregulated by 4-fold. Interesting, that's photorespiration. So we put too much light onto the system and photorespiration starts occurring. So this is where our fluorometry can get information from the proteins now. So we can start to see that there's regulation occurring. Rubisco, we've got Rubisco increasing significantly. We've got ferredoxin reductase increasing also. Of the new proteins, that is the new proteins that aren't in the control gel, we've got glutathione metabolism, which is a reactive oxygen species. We've got HSP70s, we've got chaperones. It means that we've got a lot of damage happening in the photosystems. So the proteins are responding in a way to protect the plant from high light. Here's the low light one, same deal, the two different. So this is the low light gel, this is the control gel. You can see there's a huge difference. And what's happened is a lot of downregulation because there's a lot of dots that aren't up here. So now we've only got 40 differential proteins for low light, and there's the metabolism. there's photosynthesis, there's reactive oxygen, and there's the protection, the antioxidant system. So there's not a lot of need for proteins to protect yourself from low light. And look around the top here, most of these proteins are all downregulated. There's not much upregulation of anything. There's not many new proteins. I won't run through what's happening there. So now I've brought those two back together. So this is the sun one, this is the low light one, and you can see that you've got a lot of changes in the proteins, whereas this is all downregulated. And this is the point, this is the energy metabolism. There's the photosynthesis in the low light, but the same 24% is happening with antioxidants. So it's quite a shift in how it responds from low light to high light. This is a nightmare. This is where we've got cartoons coming in. Now this is the problem. I've had to bring, or Manage has brought all of this data together. So red equals regulated proteins, yellow is new proteins, up and down arrow. So all I'm going to draw your attention, this is the highlight. Look down the bottom here. Reactive oxygen, you've got a dozen new proteins, and they're new because they've got yellow dots beside them, and they're all red. So reactive oxygen is one of the most active parts that's responded. Photosystems are responding. There's responses all over the metabolism to high light. Low light is a much calmer image, and there's a lot less detail. Photosystems are downregulated. Reactive oxygen is downregulated. There's not as much happening in the low light proteome. But we don't have pathways. We've just got protein regulation. We can't map that onto anything. So this is where we come into metabolomics. So metabolomics is now getting us close to a phenotype. It's describing things that we actually understand. So sugars, amino acids, alkaloids, phenolics, sterols, all of these compounds that make sense comes through the metabolomics. So we run a number of different techniques, so it can be non-targeted, targeted, or profiling. Most of the data I'm going to be showing you is non-targeted because we're looking for global metabolomes. Just quickly what you've got to run through, and I like this slide, you actually got to talk to people before you run your experiment because one of the biggest problems is you run your experiment and then you say, why didn't I do this with my replicates or something? So talk to the experimental group before. You do your sample prep, you run through the mass specs of the NMR, data processing, you go through libraries again, you come up with your KEGG maps, you build your hypothesis from what you've seen, and then you go back and you start your experimental design again. So this is the way that we're hopefully going to be moving into the future. So the metabolomics is really dependent upon having excellent databases, and we don't have excellent databases. So these are master charge data archives. So for all of the busted up compounds, you need to actually be able to identify them. You need to process, so noise filtering, baseline correction, this is all chemistry. For higher plants, you've got about 500,000 identified metabolites. For aquatic plants, you've got about 500, and I don't know why they're all Laurentia, they're not even, a seagrass. So we're struggling that we're identifying metabolites against higher plant systems. We don't know, we don't have standards in seagrasses, and that's a big problem. As I mentioned before, we're going to be moving into doing fluxomics, which is looking at metabolic fluxes of the metabolites using stable isotopes, and then the KEGG pathways is the way that we come up with our pictures. So Manager's got us to the point now where we've got some very interesting new metabolite profiles. We're running a GC-GCMS TOF mass spec, and that's giving us thousands of identified proteins. But we're coming back to using the higher plant database where we've got about 200 that make sense. So this is helping us now to put this layer into the system. 5, okay. This is the eutrophication experiment. This is ammonia. Just quickly, so this is controlled 200 microeinsteins, low light, sticking 5 ppm ammonia in and 6 ppm ammonia. We've got photosynthetic pigments, all the rest of it. All I'm going to talk about is the proteome and the metabolome. This is informing how we bring this data together. There's the gels. The circles are downregulated, the green are new proteins, and the squares are upregulated. We've done the same thing with ammonia. They're the same patterns you've seen before. This is the new information. So this is the metabolite. Now, if you read down the side here, secondary metabolites, organic acids, sugar derivatives, sugars, amino acids. That's all of the data we've got from controls, low levels of ammonia, high levels of ammonia. We bring this now together using the KEGG maps. So we've now got gene regulation, we've got proteins, and we've got metabolites. We can put the metabolites onto the structure of the pathway and understand how the interaction's occurring. What we have to do next is describe the patterns and understand where the significant patterns are. This is network analysis, and what we've got here is we've got our amino acids as the endpoints, and we've got the connectors back to find which of the metabolites are actually co-eluting. And so once we've identified which of these particular metabolites are important, we'll put it into a 96 well plate, and we'll have, fatty acids, oxylipins, sugars. We'll have different compounds across the 96 well plate. We'll have a healthy seagrass and a stressed seagrass, and this will give us a metabolic test. We can actually understand what's changed in the seagrass. I'm not discrediting, or I'm still excited about what Kramer's work's going to do, but I think that's going to be the future. So the toolkits that we hope to have operational, our original Gladstone one, that took a lot of work, and we'd have to take a single data, run 2 samples with a dredge and a control, 3 biological reps testing 15 genes. Now we can run 30,000 samples using the nanostring, or 1 hour to do the same thing. I've shown you what we're going to do with the metabolite testing, and I think the other parts of the group, we're going to develop biosensors using ammonia and copper, hopefully, based on Nassim's work. There's an awful lot of stuff that I've talked about today, which is not stuff that I've done. It's this broader group. So you've seen Matthew, you've seen Manoj, Uni didn't have an image up. This is a bunch of the current PhD students. Casper's in the audience. Some of his data has helped inform Peter Davies up at JCU. Mike Rashid and Gabriel Pocaccini have helped as well. There's a whole bunch of funding agencies that have helped. I've got a quick plug for a book that's coming out, when's the 19th? Like about next week or something. So Gary, Tony, Larkin and myself have been editors on this delightful challenge. Gary and I had hair before it started, and look what's happened. So this will be coming out, and you can see all of the chapters, and there's probably a lot of people in the audience that have contributed. That's the end. So thank you very much for your time.
Thank you, Peter. I'm sure you all have questions, so we're going to leave about 8 to 10 minutes for question time, and hopefully none of you are too hungry. So do we have a first question from the floor? Yes, Sven.
Many of the results that you've shown are for Zoster Muelleri.
Yes, absolutely.
And I wonder if you think that this is a general phenomenon for seagrasses, or is it true for terrestrial plants, or are we in the forefront now with seagrasses on all this?
No, I'd love to think that we're in the forefront. I'd hate to suggest that it's common across even the Zosters. Considering the problems that we had going from Zoster marina to Zoster muelleri, slight changes. So I think the general process may be correct, but it's going to take a bit of work to validate it with marina. As far as, I think we're all excited by the quirkiness of seagrasses, and they have a lot to offer, and there's a lot of exciting biochemistry that's happening here that may not be happening in higher plants. So yes, I think we need to be aware of it, and we need to exploit it, but we've got to not gild the lily too much and say that the world centres around seagrasses and they've got these incredible things. So we could be informed, and there could be useful things for crop plants, I hope.
Anybody else has a question?
Your in silico approach to seagrass biology is very interesting, and in the whole of your presentation you are mainly mentioning on the coding part of the genes from the genome.
Yep.
Which is very minimal, as you know. But what about the tandem repeats of the satellite DNA? there is no any mention of that.
That's one of the challenges of looking at populations. And so we're increasingly trying to make sure that our sampling is wider. And yeah, I think that's an important challenge. And I think Mitch is completely aware of it. And I think, yes, it's an opportunity for doing more research in this space. Yep.
Then, when I was seeing about the pattern recognition,
Yeah.
any pattern has been recognized from seagrass.
What type of pattern?
In proteomics, you are speaking about pattern recognition.
Yeah.
Any specific patterns has been recognized in the physiology or any other aspects of seagrass biology?
At this stage, I'd say no, but I think it's going to take the broader community to be more aware of it to find the patterns. So I think what we're looking for is the tools to allow people to do it, because I don't want to see a whole bunch of cartoons up on slides for the next 5 years. I want us to go beyond cartoons and to actually have empirical evidence for particular co-relationships. That's what I'd like us to move forward to.
Any other questions? Oh. You guys are just making me run now.
Thanks, Peter. It's really exciting to see these advances, and I was just reflecting on a time you and I were sitting at a coffee shop talking about these things when we were both fresh from our PhDs, and just how far things have come, and particularly in the last couple of years with the reduction in costs and the new tools. A couple of questions. One is, when you chose the material to do your transcriptomics, etc., are you just using local material, or are you using material that you know something a little bit more about, or what?
Okay, good question. That one, that comes down to population differences. So the first work that we did was at Gladstone, then we did work down in Marabune Lakes.
Yeah, okay.
So they're transportable. Our primers that we were using did work there. However, at Gladstone, populations across Gladstone were enough to make things a little bit complex.
So what's really interesting around the New South Wales Victoria coastline, as if anyone goes to Coors Talk tomorrow, it's a transition zone between two lineages, and they're mixed up, and you could really easily have very different signals.
Absolutely.
Just because you've got and that's exciting in itself, but for this kind of work, it's a word of warning. And if I was you, I would have on your second slide, where you have the first set of your 6 boxes. Before genomics, there's got to be a box which is genetic system, which is about that kind of history. And it's for seagrasses in particular, as well as some of these other old lineages that have incredibly deep histories. We're finding that evolutionary history, the insights we get from the genomics are that they're much more complex than we ever anticipated.
Agreed.
Yeah, the second thing is the exciting move towards understanding the phenome, the phenotypic expression of all of this, not only genetic signal, but the environmental parameters which you outlined today, are starting to get to us being able to really understand those mechanistic and synergistic outcomes from changing environments. I guess my question is, how do we jump from these model type systems? So although seagrasses are non-model organisms, we're now using the Zoster as a model, to some, I think this is what Sven was getting at, some more generic statements about seagrass systems. Do you anticipate needing to do across lineage comparisons, or do we need to do more at, say, the population scale, or do we need to scale up? I mean, you can go either way, but what do you think the next step is?
I suppose I agree with both. The scaling, the population are critically important. The point that you didn't mention, and the thing that I've been most frustrated is I work closely with people in Syro, wheat and rice, and whenever I present this kind of stuff to them, they go, well, where's your knockouts? You know, where's your crosses? And we did try 2 years ago to get a student to start doing exeniculture, to start having lines of seagrasses that we could actually breed and grow and have knockouts. So that's another part to it. And I kind of think it's, I don't know, I think it's probably not worth it for seagrasses to get that level of detail. Oh, we've got Mitch's response to it.
I think for some things we have to do it. I know Paul Lavery's group at ECU did some tissue culturing work with Halophilus so that we could start to explore some of those issues, and in fact tissue culture may be a better achievable approach to getting individuals into growing conditions, and then at least you have clonal replication. But I think the other part of it is, and one of the missing parts that Gary would probably agree with me on, is understanding the seed germination biology enough so that we can do these experiments is a critical part of our missing understanding of the biology. And I know he's got people working on it, and there's people around the world working on it, but our ability to do this kind of work is underpinned by that relatively weak understanding of some aspects of biology which frustrate us all. But this kind of work tells us it's worth investing in doing some of it. Maybe it's not worth investing in you guys trying to do it right now, but someone in the future needs to make these tools available.
Katie Chartrand's working with us on the seeds as well.
Yeah.
So we are starting in a small way, but it's outside.
Anyone? Rob?
So Peter and George are talking enormously, they're really great. But in the theme of the conference, translating this to management, so I'll come back to you and say, okay, we've got a dredging campaign coming up in Gladstone. Dredges are worth $2 million a day. They have a window, they come out from overseas, at the end of that window they go home. Are these techniques going to be, are you confident, given the complexities of species in a place like Gladstone and the issues of senescent periods and tide changes, light changes, are you going to be confident that we could say to a dredging company, okay, stop, because these techniques have shown us that the plants are at a point where they're not going to survive if you keep going?
Yeah, absolutely, absolutely. So our first data set, I'm confident that we picked genes that showed trajectories that told us exactly what we needed. I think the weakness with that is that we had so few genes to work on. Now with the nanostring, 50 photosystem genes that we go back, resample, and I think Mitch's point and your point is we have to have it Gladstone-centric, and we have to make sure we understand the population genetics of Gladstone. Then absolutely, I think it should be a technique, and it's going to be even cheaper than it was before, now with the nanostring. So quicker, easier, and more accurate.
Paul?
Thanks, Peter. Great talk. This might sound like a really stupid question, but following on from Rob's question, do the responses differ depending on which part of the plant you're going to work with? So if you're trying to translate this into a field monitoring program, for example, the PAM work, depending on which part of the leaf you pick, you'll get a very different response. Is it possible that there's heterogeneity within the leaf or the seagrass plant?
We've looked at some of that, and I think we can generalise it well, so that provided it's a growing leaf, it's robust. So no, it's not. There's not sufficient variation across the leaf blades in these responses. So between populations, yes. Within a rammet, no.
I think we have time for one last question.
I've always been really intrigued by this lack of an ethylene sensor in the seagrass that was found when the genome was analysed. And I hear from people that work on rice and water plants how important ethylene is in the breeding and everything. And this has a function in stem elongation. Well, seagrasses have got stems, so why don't they have an ethylene sensor gene? Why would it be knocked out?
So we need to do a lot more in this space, but rice exfoliates when it senses flooding. So it creates erenchyma and it extends, whereas seagrasses have got erenchyma and they're already linear. So there's no need for the seagrasses to change their morphology the same way that rice does. Equally, a gas isn't the most effective hormone to be moving around a permanently submerged plant. So I think what we're going to find is alternate hormones that do the similar processes, but it's not a gas-based hormone. So I think what we've got to be looking for is hormones and regulatory combinations that correlate with what ethylene should have done, but it's being driven by something else. So I think there's a smoking gun there. I don't know where it is, I don't know what hormone it is, but absolutely the processes that, apart from stomata control, you need something to replace ethylene.
Thank you, Peter. I think you've given us a lot to think about. Peter will be here until Thursday, so feel free to accost or approach him during any of the breaks. Would you all like to join me in thanking Peter Ralph? Thank you very much.